Next, we're going to jump into our guest presentations. As Jennifer mentioned, this is a series of presentations laid out to give us some ideas about the background on red tide and also some concrete actions that can be taken to mitigate the harmful effects. The first one is going to be an overview of red tide bloom dynamics presented by Dr. Kate Hubbard of the FWC Fish and Wildlife Research Institute. The purpose of this is to provide a briefing on the latest red tide and harmful algal bloom, or HAB, research and present a new suite of tools which can be used to better understand bloom dynamics and increase relevant observations. The CCMP connection relates to Water Quality Improvement Action 5, reducing HABs, specifically activity 5.1, supporting HAB research and monitoring and measures to reduce the environmental, social, and economic impacts through the identification and reduction of anthropogenic influences. With that, welcome Dr. Hubbard. Please go ahead.
Thank you so much for the opportunity to speak today. And Jennifer gave a great overview. And we've also have an excellent set of speakers today that will be touching upon very important parts of red tide bloom dynamics. And so I thought I would give a quick overview, touch on some of the critical research and monitoring tools. And I just wanted to thank our team at FWC, but also the numerous partners involved with red tide monitoring. And that includes many of the people on this meeting or in this meeting today. Next slide, please. So there are several different marine HABs in Florida that present human health risks. Karenia brevis is one of those. It produces a suite of brevitoxins. These are neurotoxins, and they can produce neurotoxic shellfish poisoning if they are consumed. The typical vector for that is shellfish. And then they also can produce respiratory irritation when those toxins are released from the cells. That's generally during wave action at the beach and is especially bad during onshore winds. I won't talk about the other HABs that we have, but a number of these are important in the Charlotte Harbor estuary. Next slide, please. So to give an overview of bloom dynamics first, if we look at this map, it's showing the different sampling locations where red tide has been detected going back to 1953, which is the beginning of FWC's record of these events. And we know that initiation typically occurs in the late summer and the fall in Southwest Florida. And if we look at that particular area, you see that there are a lot more dots there. And we've had at least 57 blooms over the past 66 years. And so there's a bloom there almost every year. If we look at some of the other regions across Florida, the Panhandle, we started observing blooms there later than in Southwest Florida. And we see that roughly 30% of the time there's a bloom there. And that sometimes the blooms in Southwest Florida are transported to the south, impacting the Florida Keys. And then in rarer cases, they can be transported all the way around to the east coast of Florida. In terms of what constitutes a bloom, there are multiple factors involved. I'll go over some of those today, but we often think about advection, which is simply moving cells around. That can be really important for both concentrating or dispersing cells in the marine environment, but also growth. And I'll touch upon some of the things that are really important for Karenia brevis or red tide growth. In terms of termination, what causes a bloom to end? This is something that we're still really actively investigating, but there, again, physical forcing is really important. So the ocean currents, as well as a number of other potential loss factors, which again, we're still trying to get a better handle on. Next slide, please. So I mentioned this initiation that occurs in the late summer or the fall. And we have a hypothesized bloom initiation zone that sits offshore, and it expands from off of the Big Bend region south, encompassing the greater Tampa Bay region, and then also further south to roughly offshore of Sanibel. And that is really important because the deep ocean is where these blooms initiate, and those currents are really important for pushing the cells towards shore. Next slide. And just to give another depiction of this, this is sort of a 3D view of the coast. And so when we have those cells that are starting offshore, we have the ocean currents that are transporting them near shore up onto the shelf. And we have surface winds that are involved as well. And next slide. And then we start to see these blooms occurring during periods of intermittent upwelling. And that's simply when water is advected from the deep ocean up into the surface ocean. And once that happens, it can really help concentrate the cells near shore. It can also help push them to the surface. When there's intermittent upwelling, there can be, when that process kind of shuts down, it can help retain those cells and again, concentrate them in the near shore environment. And that's really important because there are not a lot of ocean nutrients offshore in the Gulf of Mexico. There are some exceptions to that, but it's really important when the bloom does come inshore, when cells do come inshore, they can use both ocean and land-based nutrients to grow. So much like plants, they require a suite of nutrients to be able to grow. Next slide, please. So just to give a little bit more context for this organism, some of the earliest reports, there are reports of fish kills in discolored water going back to some of the Spanish explorers. Whether or not those were caused by red tide, we start to have reports that include things like smells. So if you've experienced respiratory irritation, you know that the blooms can have a particular odor. Some people are more sensitive than that to others. And then also widely documented marine mammal and other wildlife mortality events. And so those reports were really in Florida, started in the early 1800s. And from those reports, they describe really severe blooms interspersed with fairly regular blooms. And that's fairly similar to what we're seeing today. One of the reasons that we see blooms in Southwest Florida is because we have a nice wide shelf. And so that really helps with that process of moving cells from offshore to onshore and also retaining them. And Florida is not the only place that experiences regular blooms. They also occur along the Yucatan shelf. And if you look down, you see that there's a similarly wide shelf there. Texas experiences blooms much less frequently. And that's because they're advected north from Mexico, but that doesn't occur the same way in every year. So it's a little bit of a different process over on the Western Gulf. The blooms that occur in Florida can be transported north and sometimes impact other Gulf states like Mississippi and Alabama. And then rarely, as I mentioned earlier, blooms can be transported up the east coast of Florida and then also further north up the east coast of the United States. So blooms can move around quite a bit. However, this organism does seem to be endemic to the Gulf of Mexico, and we haven't experienced bloom initiation in other regions other than the Gulf of Mexico. Next slide, please. So some of the longest documented red tide blooms are shown here in this image on the left. And blooms, the longest bloom that we've observed lasted 30 months, and that was from 1994 to 1997. I think probably most of you are familiar with some of the blooms that have occurred recently, especially the 27 to 2019 bloom. And then we just had the 2020 to 2021 bloom wrap up. Those blooms lasted a year or more and caused devastating consequences. And if we look at the number of years with a documented bloom by month since 1953, we see that in the late summer and the fall, that's when we typically start to see more years with a bloom. That's when we start to see the onset of the event. And that's also at the same time, we have the end of the wet season. And we also have the peak in hurricane activity or tropical storm activity. So lots of things going on in that time that sort of complicates our ability to definitively predict when a bloom might start and then what might happen once we have a bloom, given that there's a lot of different things going on with the ocean. Next slide, please. So I mentioned those two recent events. The 2017 to 2019 event, it impacted 22 different coastal counties, some quite persistently, and it definitely caused a lot of impacts in the Charlotte Harbor area, Tampa Bay area. It also did get advected to the east coast of Florida and caused impacts in the Panhandle. I'm showing maps. These are monthly summary maps showing our cell count data. So anything that's red or orange indicates a bloom, so over 100,000 cells per liter of red tide. And you can see the different distribution patterns in October. Those were really, those months were really when the bloom was quite severe during both of those particular events. On the left, or excuse me, on the right, we have the 2020 to 2021 event. And that impacted 17 coastal counties, and those were all on Florida's west coast. It did cause impacts along the Big Bend region, which is somewhat unusual. Next slide, please. So during long-lasting blooms that last over a year, so they, start to persist into the time where we start to see new bloom initiation. One of the questions that we have then is whether we have simply advection of the persistent bloom or new initiation. And that was certainly something we were wondering about this particular year, and it's something that we'll look into more with some of the modeling. But if we look at that going back to the 2018, the year in 2018, the bloom then, we did do modeling. This is a paper that came out of Bob Weisberg's group. And what we can do with this is look at the bottom currents, and we can use oceanographic circulation models, and we can basically hindcast these events, and we can see if the circulation was conducive for pushing new cells on shore. And with this, we see the start date being in August, and then these trajectories were allowed, or excuse me, these models were allowed to run for a few months into October. And with that, we can see that, in fact, the transport pathways that year were conducive for bringing new cells on shore. So whether or not we had new initiation that year is still an open-ended question. We had a lot of storm activity as well. And it's most likely that there was the potential for there to be both advection as well as new initiation. Next slide, please. We can study that further using tools like ocean gliders. These are instruments loaded with sensors, and they look a little bit like a torpedo. They move up and down in the water column. When they surface, they send their sensor data back to us on shore so that we can generate profiles like this. And this is showing chlorophyll. It's a good proxy for algae. We can see some of those subsurface peaks of chlorophyll over the course of the deployment. And this was deployed for almost a month. And sometimes you can see those subsurface peaks sort of start to move up a little bit. And this is, of course, as the glider's moving near and off of the closer and farther away from the shore. But some of those peaks where we start to see it move up in the water column, those occur when we have some larger storms go through. And we had Tropical Storm Gordon, Hurricane Florence, and Hurricane Michael. And those were really important for, again, moving the bloom around in 2018. When we went to go retrieve the glider, we did find that that subsurface chlorophyll area shown at the very end on the right-hand side of that plot, it did have red tide present. So we're using these types of tools to be able to go out and direct our vessel-based sampling to get a better handle on what might be out there, and again, try to better understand some of the critical questions involving bloom dynamics. One other thing, since I mentioned nutrients earlier, we have now a nitrate sensor, and that's a royal we because USF and MOTE are in charge of glider operations for our different red tide programs. But with our USF program, we do have a nitrate sensor now that we deployed successfully this past year to get a better sense on what the nutrient conditions offshore are as well. Next. So one of the questions that we have that we're working on is the life cycle of Karenia brevis. So this is showing, if you look in the upper left-hand corner, you see asexual division. This is the most important process for growth, and this is what's going to be happening during a bloom. However, there on the left or on the right-hand side, we have the sexual cycle, and then in the lower left-hand corner, we have sort of an unknown stage, which is still to be described, and that would be a resting stage. Next slide, please. This is something that we haven't yet defined for Karenia brevis, but recent research has shown that closely related species, Karenia mikimotoi, which exists globally, not just in the Gulf of Mexico, does have a resting stage. That was the first set of research, so just finding it and defining it was interesting. And then recently, this past year, another study came out that found that there's a 50-year record of this resting stage in the coastal sediments off of the coast of China. So basically, they were able to detect Karenia mikimotoi going back to the 1970s. And I just show some images of what that resting stage looks like. It's pretty nondescript. I don't know that I'd be able to pull it out of a sediment sample that we would pick up, but what we can use and what they used are a combination of microscopy with probes that are specific to the organism, as well as genetic methods like quantitative PCR. And one of the good things is that we have all of these approaches in hand already for Karenia brevis, and we're working with partners both in the state and then also internationally to try to get a better handle on whether or not Karenia brevis also has a resting stage. And again, this is really important for being able to better constrain bloom initiation, when and where it might happen, but also where these beds of cysts may reside offshore. Next. We also know that Karenia has a flexible morphology. When cells start to become stressed, we do see these circular cells, sometimes referred to as balls. Some of the work that's been done in Alabama, where salinity is typically lower than what we see on the West Florida shelf, has shown that at low salinity, these stressed cells end up forming circular morphologies. And we've been doing work at FWRI to try to get a better handle on that too, and whether that's a precursor to forming those resting stages. And so we can look at that. And if you click again, please, these are images of cells that we can generate using different imaging cytometers. So basically they can query all of the different cells in a sample that we feed it, and then we can use different algorithms to sort those out. And one of the tools that we've been using quite a bit is the imaging flow cytobot. This is shown there in just kind of it's a cylinder, but it does a whole lot. Mike Parsons at FGCU has one also, and we have several. And what we can do is deploy these on a ship. We can put them in the water and leave them for a while. Or we can run them in the lab. And what we can do is identify single cells. We can identify those spherical or stress cells, but we can also identify paired cells shown at the bottom. And that's really important because that tells us that Karenia brevis is actively dividing. And when we looked both in 2018 and in 2021 during the peak of those blooms, we were able to see the transition from intensive division periods into those more stressed timeframes. And that's really helpful for, again, getting a better sense of what's going on with these cells and potentially then connecting it to other dynamics so that we can understand some of the drivers. Next slide, please. So as I mentioned, the asexual division, that's really important for these blooms. Karenia brevis is actually a pretty slow grower compared to a lot of other phytoplankton. That might be surprising given how prolific it can be. But this phytoplankton, based on some of the research that we've done in the lab, and this is again the royal we know that in terms of light, nutrients, temperature, and salinity, it's fairly flexible. I'll go into detail on the first two just to give a little bit more context there. Next, please. So these are some experiments. That's how we grow cells in the lab on the left. And we have LED lights, which is something that we've been able to integrate into our culturing. And we grow Karenia brevis in the lab to see how it responds to different environmental cues. The LED lights have been great because they're cool. Usually in the past, when we've tried to have really high light simulating what's going on at the surface ocean, we would also be heating the cells up because the lights are producing so much heat. And doing this, we were able to simulate a huge range of different light levels, matching different levels that we might expect in the environment. And those are shown in terms of the responses or the cells per mil. We see that Karenia is able to grow at all of those different light levels, with the highest light levels being in the reddish colors, lower light levels being in blue. So we do see that they prefer high light. And then also we see that that response across different strains is pretty consistent. And that's something that is a little bit in contrast to what was previously thought, where we used to think that Karenia brevis really thrived at low light, sort of what we would expect down deep in the ocean. So again, this is really important for bloom development. These organisms swim up and down in the water column as well daily. So this allows them to access a different suite of light levels. And so now we know that they're able to grow at all of those light levels. So this is one way that they're able to really, thrive in the environment. Next, please. We also know from some of the research looking at nutrients that Karenia brevis can uptake a number of different forms of nutrients. These may have different origins from, again, land-based or ocean-based sources, and they may also be biological sources like DNA or RNA. So this is looking at phosphorus, and again with cultures in the lab, and we see that although there is higher growth on some forms, this organism was able to sustain growth on 29 different types of phosphorus. Next. And we also know from a lot of the research that's been done that Karenia brevis has a similar propensity for growth on different nitrogen sources. And this has been examined in a number of studies, but one in particular looked at more than 8 different sources or types and was able to see that there was a strong preference for ammonium, then nitrate, then urea, and then some other sources that are likely to be present in the environment. Also, it appears that Karenia brevis is able to modulate that nitrogen uptake. So again, not really good news for us. Karenia brevis is able to use a lot of these different sources. And when we look at nitrogen and phosphate and where those are coming from, there's a lot of different places in the environment that could be coming from, originating. And you'll hear a little bit more about that with some of the other speakers later today. And one thing to be aware of is that there's not a lot of nitrate out in the ocean. So their preference for ammonium and their prevalence for being able to use a lot of different sources, it likely has evolved over time because of that. Next, please. So as we start to think about the complexity of bloom dynamics, these factors that are really important in the growth of Karenia brevis, they help it prevail over a number of different conditions lasting across a year. If you think about the temperature or salinity conditions as this organism is moving from the deep ocean into our estuaries and also persisting for over a year, sometimes up to 30 months, it's able to thrive across a really broad range of temperature or salinity conditions. That said, we generally don't see Karenia much above 30 degrees Celsius, and we also don't see Karenia very frequently below 22 PSU. So there are some limits. It's not, infallible, but it's definitely very versatile. Next. So if we think about predicting Karenia brevis, right now we have short-term forecasts that are really tied to the ocean circulation. And as we start to think about forecasting or understanding these blooms at seasonal to interannual timescales, which is something that we're very interested in, again, trying to connect some of these potential drivers with what's actually going on in the ocean. We know that the loop current offshore, shown on the left, is really important for setting up circulation patterns on the West Florida shelf. And if we look at the right, that's showing the loop current in red as it moves around and different patterns that have occurred over a 25-year period. These are the most common patterns. And we have upwelling near the dry Tortugas because of the forcing there in those boxes that are outlined in pink. And we have looked across time. One of the things that we see is that when there's too much upwelling that occurs, we don't typically tend to have Karenia brevis blooms, or they tend to be less severe. And particularly in that region near the dried Tortugas, which is referred to as the pressure point. And when we have the intermittent upwelling, that's when we typically do have blooms. And so this is something that we're trying to use to help advance our seasonal forecasting so that as we're moving into a new bloom season, we can get a better sense for what might happen next, given the conditions going on, with the coastal circulation. Of course, there's a lot of other factors that come into play. I haven't touched on some of the biological conditions that may cause blooms to thrive, other than perhaps life cycle dynamics, but we also know that brevetoxins may be a deterrent to some grazers, things that would potentially eat Karenia brevis, and so that may be another mechanism that allows it to thrive so widely across Florida. Next, please. I just wanted to wrap up that we have new questions as we move through thinking about bloom dynamics, the initiation, what sustains a bloom, what might terminate a bloom. And then we also have some of these new tools like gliders coupled with nitrogen sensors, the imaging tools that I mentioned that can help us evolve our hypotheses. We also recognize that these tools and integrated observation networks are really critical to continuing to advance the science for Karenia brevis. I just wanted to also show, there are other algae that impact Charlotte Harbor. This past year, we had reports of there being a really bad red tide event in Charlotte Harbor, after there was already a really bad red tide event in Charlotte Harbor. And in fact, that water discoloration that you can see on the left was actually due to another species of dinoflagellate, Tripos hircus. So it's just, something to look into in the future. And again, algae are always surprising, and they're able to respond to such a different variety of environmental conditions. So that's partly why having these integrated observation networks is really so critical. And I'll stop there.
Great, thank you so much, Kate. Is there any member questions?
Sure, Corey, I have a question for Kate. This is my Kate, when you show the potential for a resting stage and the salinity with the 20 ppt salinity threshold, and you mentioned doing some sediment analysis on offshore seabed to look for resting stages. Would you look also onshore, because that's where we would look at those type of salinities, or how would that work? How stress would look like offshore so that it would create resting stages, because offshore the salinity will not go down to 20 PSU of salinity.
Yeah, so that's a great question. And we are looking both near shore and offshore. We are also trying to find the best places to look in terms of where offshore there's most likely to be these types of reservoirs, just based on the sediment itself, because there are different types of sediment that are more likely to harbor biology like this and promote it. In terms of what might happen in the sequence of events, once we have a bloom start to peter out, we start to see these stress cells. And again, this is a working hypothesis, but they may be more subject to being transported around by ocean currents. That means that they may be deposited in nearshore environments, but they may also be pushed offshore. And we do know that dispersal along the coast and movement along the coast is something that we tend to see at the end of a bloom. But again, there are a number of different cases that that we can see at the end of bloom. So not every bloom fits into that. But in terms of what might cause the cells to be stressed, salinity is just one, and that's most likely to be prevalent in estuaries, but there are likely to be other sources as well, of other factors that are going to be stressing out cells. And that's something that we are actively looking at. Cindy Heil and Pat Glibert and a number of other collaborators, including myself, have a 5-year project funded by NOAA to try to get a better handle on what might be going on during termination, and looking at a number of both biological, physical, and also chemical factors. So very good question. And whether or not we see the same things that they see in Alabama here, I think that's another important question, because again, they tend to have lower salinity observations in the Northern Gulf in general with respect to these blooms. And so whether or not that's a mechanism in terms of why we don't see them sustained for very long periods of time. And I suspect bloom termination is going to end up being just as complex as bloom initiation and sustenance.
Thank you.
Thank you. Any more comments or discussion?